we're in chapter 7. Let's continue chapter 7. See if we can complete this today. And there we go. So this is where I left off. This is an overview of how eukaryotes use the mitochondria and actually enzymes in the cytoplasm to extract energy from organic fuel. Mostly we track this story by looking at the fate of glucose. And it's relevant to reality because even though we don't eat that much glucose, a lot of the materials we eat can be converted into glucose or some intermediate along the pathway en route to generate ATP. We'll talk about near the end of this chapter how ATP is generated from other nutrients like fats and proteins. A couple of things here. So this process involves 3 sets of events. Glycolysis is where we'll start. This is the part that occurs in the cytoplasm outside the mitochondrion. This is, again, I mentioned that your book talks about this being one of the most ancient metabolic pathways. And the evidence for that is that all life forms perform glycolysis. That means that this chemistry was in place before cells diversified into different species, different types of living forms. All right, all using pretty much the same cellular machinery, the same enzymes in this case. Glycolysis, sugar splitting, is a very inefficient pathway. It's anaerobic, no oxygen required. In fact, the chemistry was in place before oxygen was available in the atmosphere for creatures like ourselves. And it's inefficient in terms of how much ATP yield you recover from glycolysis. Per sugar, your net ATP production is 2. Okay? In the process, glycolysis, the 6-carbon glucose, pardon me, is split into 2 3-carbon products called pyruvate or pyruvic acid. Okay? And in this 3-carbon product of glycolysis, there's enough energy remaining to produce another roughly 28 to 30 more ATP. So it's a very inefficient process. It is fast. Okay, we'll talk about how some of our tissues do perform glycolysis, like especially skeletal muscles, because it's fast and it can produce ATP quickly, but the yield is poor, as I mentioned. Now, in the production of ATP, it's accomplished by something called substrate level phosphorylation. I'm going to jump to the next slide and then come back to this. Substrate phosphorylation involves the direct transfer of a high-energy phosphate to ADP. And it's catalyzed by an enzyme. And what kind of enzyme would this be that adds a phosphate to a target molecule?
Kinase.
It'd be a kinase. It's a kinase enzyme. Okay? We'll see that this type of ATP generation, substrate level phosphorylation, occurs in glycolysis and in the citric acid cycle. Okay? All right. The direct transfer of a high-energy phosphate to ADP, catalyzed by a kinase enzyme. Okay. You see it happens here, and it happens in the Krebs cycle. Okay? But the bulk of ATP produced in aerobic respiration is through a different mechanism. It's called oxidative phosphorylation. Or you can think of it as chemiosmosis. Now, we know what osmosis means. Osmosis refers to diffusion of water from high water content to low water content. Okay? In this case, it's the diffusion of protons across a membrane, across this inner membrane here. Okay? We'll look at the mitochondria, but there's an intermembrane space and then this inner region here called the matrix. And these protons will diffuse from this intermembrane region to ATP synthase into the matrix. And that movement of these charged ions down a concentration voltage gradient provides the energy to fill the bulk of ATP. This is where the bulk of ATP is produced in this unique mechanism. Okay? And we'll talk about this amazing pump, ATP synthase, this rotor. It's a very ancient machine. Almost all life forms have this machine. And you can use it to make ATP, or you can run it in reverse, use ATP energy to power other processes. Okay, that's how bacteria swim. They use they use ATP-powered ATP synthase to actually propel a flagellum. All right, so it's a very ancient machine there. Okay, so there's the bulk of the payoff, and we'll look at the process here. Of now, I'm sorry, I referred to the osmosis portion. Protons diffusing down an electrochemical gradient through this ATP synthase rotor. And that's what the energy is to build the bulk of ATP, and that's what the chemie refers to as forming this bond. Okay? High energy phosphates attached to ADP. So it's a chemical reaction powered by an osmotic or diffusion of protons from the intermembrane space into the matrix. All right. Before we get there, though, this osmotic process, that is a gradient of protons, right, a high concentration of protons diffusing inward, that gradient of protons is created by an electron transfer system or electron transport chain that's embedded in the membrane, and the electrons that flow through this transport chain to power the gradient generation of protons. I'm saying a lot of things that may not make sense. That's derived from the oxidation of something called acetyl-CoA in the citric acid. So here's really the centerpiece of aerobic respiration. Okay, it's converting this molecule. Okay, acetyl is a 2-carbon molecule derived from the 3-carbon pyruvate, so it's oxidized as it enters the mitochondrion. And this is where the bulk of the energy is. 2 carbons left out of the pyruvate. It's attached to a high-energy carrier that shuttles it into this cycle. And this represents 8 reactions. It's called a cycle because the acetyl is transferred from CoA to a receptor molecule, something called oxaloacetate. And it goes through a series of reactions. In the process, essentially, the 2-carbon acetyl is stripped of all its high-energy electrons. They're all stripped away, transferred to carriers, activated carriers. Okay? Activated carriers are key to this process. ATP is an activated carrier. It carries high energy in the form of a phosphate bond. Okay? These are activated carriers. NADH and FADH2, they carry high energy electrons. Okay? CoA, coenzyme A, is an activated carrier. It forms a high energy bond with acetyl, shuttles it into the reactions inside the matrix, and then when it's released, it provides power for this reaction to occur. So a lot of activated carriers. And what's interesting about these activated carriers, they're all based on RNA molecules. Maybe they're a relic of that early RNA world where the energy systems evolved before DNA was in place. Okay? And so the acetyl is stripped of its high energy electrons. At the end of the cycle, all that's left is the carbons here. They're now in their lowest energy state as CO2, and they just diffuse out back to the atmosphere. So this really completely completes the dismantling of the sugar, such that at the end of the Krebs cycle or citric acid cycle, same thing, the carbons completely oxidized down to CO2, nothing left. But the harvest of this cycle is the high energy electrons derived from the sugar on these activated carriers. And they're the ones that deliver the electrons to the transport chain to power this chemiosmosis. Okay? And ultimately, these high energy electrons in glucose, where did their energy come from? Another topic of the next chapter. What is the source of the energy in these high energy electrons that are stripped from these sugar fragments in the citric acid cycle to power ATP synthesis? What's the ultimate source of that energy? Sunlight. Yeah, sunlight. Okay. So we got 3 sets of events. We're going to look at, we're not going to go into the complete brutal detail of each of these. That's for when you take biochemistry. We'll look at kind of the highlights of the reactions. Okay, there's plenty of detail there, but we're not going to get into the nitty-gritty details of how OH groups are replaced by a carbonyl net and stabilize the bond and blah, 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 a lot of stuff. So again, here is, we've already talked about substrate level phosphorylation. So again, this is the, again, I'll say it, a direct transfer of a high energy phosphate to ATP catalyzed by a kinase enzyme. Okay, and this is how the original version of respiration, that is anaerobic respiration, glycolysis, was able to make a low yield of ATP, very different from the chemiosmotic production of ATP at the end of this aerobic pathway. Okay, so here's the organelle. that's going to, in the matrix here, contains the enzymes to completely dismantle the sugar fragments that are imported inward, the pyruvate. Okay, so that dismantling is performed by the enzymes of the citric acid cycle or Krebs cycle. They're the same thing. They're in the matrix here. There's some other enzymes in the matrix here as well. There's some enzymes here that help to oxidize fatty acids. Because sometimes you power your body on stored fat, not carbohydrates. Okay, and so those enzymes are in here as well. They perform something called beta oxidation. We'll talk a little bit about that. All right. Now you can see the matrix where those enzymes are located. Perform the citric acid cycle and oxidize fats. When they strip the electrons away from the sugar fragments, they're transferred to those activated carriers, NADH, FADH2. They're in the matrix here. And they deliver those energy electrons to the electron transport chain embedded in this membrane here. Okay? And when you've got a lot of extra surface area here, think of it like counter space in a lab. The more counter space, the more instruments I can run chemistry reactions. So with lots of surface area here in this highly enfolded membrane, you can have millions of sets of those electron transport chains to harvest energy as fast as possible. Okay? And also embedded in this membrane is the ATP synthase that's going to depend on the proton gradient to power ATP production directly through the chemiosmotic process. Okay? This inner membrane here has special types of lipids that, in fact, these lipids are special. Most phospholipids of membranes have 2 fatty acid tails. Those of the inner membrane, they have 4, actually. Some of them have 4 fatty acid tails. That really helps to make a really